sitting biology, another video of diffusion and osmosis for the IGSC Olevels Biology. And we were covering the topic for the diffusion and the osmosis. The next topic that we have are the concentration for the turgidity and the other conditions of the cell, which is the 3.1 section of the syllabus. So let's begin with another topic that we have today, which is about the conditions of a cell when it is placed in different concentrations. So these are the four conditions in front of you. Number one is how the cells become turgid. Number second is when a cell is placed in an isotonic solution. Then we have the condition in which the cell becomes flaccid. And last we have this condition in which the cell becomes plasmolized. Let's start with the number one. Diagram number one, in which the result says that it becomes turgid. So all of these are the examples of the plant cell. Number one thing that you need to remember is that the condition of a cell are learned in the plant cell. Well, the plant cell is placed in a dilute solution when it becomes turgid. So one thing that you need to remember is when the turgidity takes place is when it is placed in a dilute solution. What is meant by the term dilute solution? The dilute solution means whenever there is around 1% solution outside the cell and approx 2% solution inside the cell. Then what happens is that the cell wall is being compressed by the cell membrane. It exerts the pressure on the cell wall, due to which it becomes turgid, giving it a very rigid structure. The concentration inside the cell is about 2%, due to which the cell membrane is pressing onto the cell wall when it becomes fully turgid. The pressure that is exerted by the cell membrane onto the cell wall results in the turgidity of the cell. This is how the cell basically becomes turgid. This results in the turgidity of the cell. Now let's talk about the diagram number two, which tells us when a condition of a cell when it is placed in an isotonic solution. So when the condition says that when it is placed in an isotonic solution, that means that there is 2% solution outside, there similarly is a 2% solution inside the cell. For example, if there is 1% solution outside the cell, there will be 1% solution inside the cell as well. In this condition, we say that the cell is placed in an isotonic solution, and thus it is in a state of equilibrium. Equilibrium means that there is no change in the state. So equilibrium means that the concentration remains same. This happens when a cell is in the same concentration of solution. We say that the cell there is no changing that take place in the cell. All right. So let's have a look at the diagram number three, in which the cell becomes flaccid. In such condition, what happens is that there is around 3% solution outside the cell, whereas around 2% solution inside the cell. What happens in such condition is that the water movement is from inside the cell to outside the cell. 2% solution means that their concentration is lower inside the cell and outside the cell, whereas 3% solution, that is, it is more amount of the solution. That is, there is a higher concentration out the cell. So what the water would be moving from the lower concentration to the higher concentration. That means inside the body of a cell to outside the body of a cell. Due to what happens is that the cell becomes flaccid. The water moves out of the cell, due to which it becomes flaccid. The fourth condition that we have is the cell becoming plasmolized. Now, why does a cell becomes plasmolized? Is when the water moves out of the cell in a larger amount. Like we have already discussed, there must be a change in the concentration. That's why the water is moving outside the cell by the process of osmosis, due to which the cell wall is pulled away. The cytoplasm is pulled away from the cell membrane. It is the condition in which we say the cell become plasmolized. We won't say that the cells shrink, but we will say that the cell do maintain its shape, but to a certain extent. When the water moves outside the body of a cell by the help of the process of osmosis, in such condition, the cytoplasm is pulled away from the cell membrane. In the process of turgid, the cytoplasm and the cell membrane exerts pressure on the cell wall, whereas in the condition of plasmolysis, the cytoplasm is pulled away from the cell wall and the cytoplasm. Such condition is known as the plasmolysis. You have already learned about the plasmolysis. Let's discuss about a small term that says deplasmolysis. What does a deplasmolysis mean? It means when the condition of plasmolysis is being reversed, then the deplasmolysis takes place. This is a small term that is mentioned in your syllabus. So you may learn the term deplasmolysis. It means the reversal of plasmolysis is known as the deplasmolysis. So these are the four conditions of a cell when it is placed in a condition where it becomes turgid, when it is placed in isotonic solution due to which it becomes an equilibrium state and the concentration remains the same, or it becomes flaccid, or it becomes plasmolized. So these are the four conditions that you need to learn in the chapter diffusion and osmosis. Let's discuss the conditions of cell in further more detail. That says hypertonic, the condition of hypertonic, the condition of isotonic, the condition of hypotonic. We have already discussed it earlier, but let's discuss with the reference to the water. The water in the hypotonic solution is moved inside to the outside of the cell. And the isotonic solution, the water equally moves in and outside the cell. Well, in the hypotonic solution, the water moves inside the cell. Let's discuss what regarding the water. How does these conditions vary cell to cell? Well, so this is a hypertonic solution. Hypertonic solution means that there is approx 3% solution outside the cell and 2% solution inside the cell, which means the water will be moving out of the cell. Water will move out of the cell. It means the water will move from the region of higher concentration to the region of lower concentration. When it comes to the isotonic solution means that the cell often becomes flaccid. It means that either there is a state of equilibrium that is established inside the solution, which means there are approximately 2% solution outside the cell, there is 2% solution inside the cell. The equal amount of water is being displaced and is entering inside the cell. What about the hypotonic solution? The hypotonic solution means, for example, if there is 2% solution inside the cell, it is about 1% solution outside the cell. Thus, the water moves from the region of lower concentration to the region of higher concentration, which means the water will be moving inside, water will move inside the cell from the outer surface, inside the cell. This is the condition of hypotonic. Let's discuss a term that is the turgidity, which is mentioned in your syllabus. What is meant by the term turgidity? Turgidity in term means when the condition in which the turgid pressure pushes the cell membrane against the, when the cell membrane is being pushed, is being pushed against the cell wall. This is the condition we say that there is a turgidity in the condition of a cell. Now, the question often arises that how does the plant cell maintain? How do plant cells maintain their rigidity? Now, the term rigidity and the term turgidity are very similar to one another. They both rely on each other. When a plant requires turgidity, it means that it needs to rely on turgidity. When the turgidity is there, it gives a structure to the plant cell, thus it becomes rigid. This is the answer to this question. Now we also need to learn about the turgid, which we have discussed in the last sheet, that says the turgid is a condition which we say that is a solid condition. Turgid is the solid condition, especially due to the high fluid content. Solid condition of a cell due to the high water content or high fluid content. Okay, all right. So the healthy plant cells are turgid, and plants rely on the turgidity to maintain their rigidity. This is the overall view of the conditions of a cell when it is placed in a hypertonic solution, when it is placed in an isotonic solution, and when it is placed in a hypotonic solution. You need to learn